Okay, so good afternoon, students. Today we are going to study the first chapter from your syllabus. This is SYBSc Botany, Paper 2, Chapter Genetics, under that chromosomal aberrations. As a general rule, the structure and number of chromosomes in an individual species remains constant. However, due to sudden changes are produced in the structure or number of chromosomes. However, due to sudden changes, some changes are produced in the structure or number of chromosomes. Such changes are called as chromosomal aberrations. Such changes are called as chromosomal aberrations. The chromosomal aberrations are of two types. The chromosomal aberrations are of two types. Number one, numerical chromosomal aberrations. This involves change in number of chromosomes, change in number of chromosomes in a diploid set, which is called as heteroploidy. The change in number of chromosomal sets is called as polyploidy. Thus, numerical chromosomal aberrations are of two types, polyploidy and heteroploidy. Second type of chromosomal aberrations are called as structural. It means that whenever there is a change, whenever there is a change in the structure of the chromosomes, it is called as structural chromosomal aberrations. It is of four types. Number one, deletion. Number two, duplication. Number three, inversion. And number four, translocation. In this diagram, you can see all the structural chromosomal aberrations at a glance. This is deletion, duplication, inversion, insertion, and translocation. Insertion is not in your syllabus. You have four types: deletion, duplication, inversion, and translocation. The structural chromosomal aberrations are of two types. Number one, intrachromosomal structural aberrations. And number two, interchromosomal structural aberrations. Intrachromosomal means the structural changes occur within a pair of homologous chromosomes. I repeat, intrachromosomal aberrations occur within a pair of homologous chromosomes. Examples are deletion, duplication, inversion. Interchromosomal aberrations occur between two pairs of homologous chromosomes or between two different non-homologous chromosomes. I repeat, interchromosomal aberrations occur between two pairs of homologous chromosomes or between two non-homologous chromosomes. Example is translocation. Dear friends, please observe the slide carefully. It is showing four types of chromosomal aberrations. Observe it carefully. Duplication, deletion, inversion, translocation. We move on to the first type of structural chromosomal aberration, that is deletion. The current slide is showing phenomenon called as deletion. You can see in the slide that during this phenomenon, a part of a chromosome is broken. Part of a chromosome is broken and lost. Broken and lost. This phenomenon is called as deletion. This phenomenon is called as deletion. This is another slide showing deletion. This is the original chromosome and the part in blue color is broken and lost. Broken and lost. This phenomenon is called as deletion. After deletion, the chromosome appears like this. Another picture showing deletion. This is a pair of homologous chromosomes, left side and right side. The left-hand side chromosome is intact. Deletion is occurring here. This part is deleted. So, after deletion, the pair occurs like this. After deletion, the pair occurs like this. If you closely observe both the diagrams, you can see that in the first diagram, the chromosome is breaking twice, here as well as here, here as well as here. Due to this double break, the middle part of the chromosome is deleted. This is called as intercalary deletion. I repeat, intercalary deletion. Whereas here, if you see the terminal part of the chromosome is deleted. So, this deletion is called as terminal deletion. I repeat, terminal deletion. Another picture showing example of terminal deletion. This example is found in human beings. In human beings, the fifth pair of chromosome appears as shown on the screen. As shown on the screen. This is the P arm or the short arm, whereas this is the Q arm or the long arm. This is a pair of homologous chromosome in human beings. This is pair number 5. This is chromosome pair number 5. Students, if you remember, last year you had done karyotype of normal male and normal female chromosomes. You had 23 chromosomes in each. Out of the 23 pairs, this is pair number 5. In this, the terminal part of the fifth chromosome, I repeat, terminal part of the fifth chromosome is deleted. Only in one of the homologous chromosomes. Only in one of the homologous chromosomes. This one, or this one. The actual picture looks like this. This part is deleted. This part is deleted. Such a chromosome is called as cry-do-chat. I repeat, human being with such a portion of deleted chromosome, the disease or syndrome is called as cry-do-chat because when this individual is born, it is said that when this individual is born, it has, whether he or she, whoever it is, has a cat-like cry. I repeat, a cat-like cry. So this is the example of terminal deletion, cry-do-chat, chromosome number 5 in human beings. This picture is showing intercalary deletion. You can see that the chromosome is breaking twice, here as well as here. If you observe closely, in the deleted region, the centromere is also present. This part is deleted. This deleted part comes out and it becomes spherical or circular in shape. Such a chromosome which is spherical or circular in shape, it's called as ring chromosome. It's called as a ring chromosome, as if the centromere is the diamond and this is the other portion of the ring. It appears like a diamond ring. So this chromosome is called as ring chromosome. This is called as intercalary deletion. The other part of the chromosome is lost. This part and this part is lost. The picture is showing formation of ring chromosome. I hope it is clear. I'll be sharing this PPT later on. We move on. So, examples of deletion are, already I've told you about cry do chat in the 5th chromosome in human beings. Apart from that, the other examples are found in maize, Drosophila, mice. And one more example in human beings, that is partial deletion of, it is a terminal deletion, partial terminal deletion of 21st chromosome, partial terminal deletion of 21st chromosome in human beings causes a disease called as leukemia or blood cancer. Leukemia or blood cancer. This type of chromosome with partial deletion was discovered in Philadelphia city of USA. In Philadelphia city of USA. Hence, this type of deletion or syndrome or chromosome is also called as Philadelphia chromosome. I repeat, Philadelphia chromosome. We conclude deletion by studying the effects. Dear students, because of deletion, the following effects are observed. Number 1, crossing over. Due to deletion, crossing over is suppressed, or rather, crossing over is suppressed in the deleted region. Crossing over is suppressed in the deleted region. Number 2, phenotype. Number 2, phenotype. Phenotype means physical character of the individual, plant or animal. Phenotype changes due to deletion, as deletion brings about loss of important genes. I repeat, due to deletion, phenotypic changes are observed due to loss or deficiency, due to loss or deficiency of important genes. Then we move on to karyotype. You already know what is a karyotype. It is the orderly arrangement of chromosomes. Karyotype is orderly arrangement of chromosomes in the descending order of its length. Karyotype changes due to structural change. Next, fertility. Ability of an individual to reproduce by sexual reproduction, that is, by formation of gametes. By formation of gametes. It's called as fertility. During formation of gametes, reductional division called as meiosis occurs. Gametes are produced due to meiosis. Due to deletion, the meiosis can be affected. The meiosis can be affected, and hence the gametes may not be produced. The gametes may not be produced properly. This reduces the fertility of an individual, plant or animal. And the last effect is viability. Viability is the lifespan of an individual. Viability means lifespan. That is how many years you live. How many years you live. Because of loss of important genes, because of loss of important genes, the lifespan or viability can be reduced. Students, with this, we complete the first type. We complete the first type of structural intrachromosomal aberrations, that is deletion. I hope it was very clear. Now we move on to the second type, that is duplication. That is duplication. Please observe the slide carefully. If you see in this slide, there are two chromosomes which are homologous. And due to this phenomenon, that is due to unequal crossing over, due to unequal crossing over, due to unequal crossing over, some genes on the chromosome are present twice. Some genes on the chromosome are present twice. If you see here, some genes on the chromosome are present twice. This is called as duplication. This is called as duplication. If you see in this diagram, it is showing symbolic genes, light blue color and lavender color. These are the two genes present originally on the chromosomes. After duplication, see what has happened. The blue gene is present twice and the lavender gene is also present twice. Examples of duplication are bar eyes in Drosophila, eyeless condition in Drosophila, vein thickening in Drosophila and hairy wings in Drosophila. I repeat, what is duplication? When a few genes are present in double doses or twice on a chromosome, it is called as duplication. It occurs due to unequal crossing over or as a result of deletion or as a result of deletion. These are the examples of duplication. Effects of duplication. Crossing over is suppressed in the duplicated region. Phenotype changes due to double dose of genes. Karyotype also changes due to structural change. Fertility, no change as the genes are not lost or reduced. Viability, also no change. So as compared to deletion, effects of duplication are mild because important genes are not lost. Because important genes are not lost.